everyone. Welcome you all to week 11 NPTEL live session, which is on telomere and aging. Okay, so like all those sessions, today also we are going to discuss few questions which will help you to do your assignments. Okay, so I think people are joining, so let's wait for 4 minutes. We'll start from 6:10. Okay, just 3 to 4 minutes, then we'll start. Okay, let's start. So till now we were discussing about different types of RNAs, right? Non-coding RNA, coding RNA. For example, if we go for central dogma of life, that is from DNA to DNA, that is replication, DNA to RNA, that is transcription, then RNA to protein, that is translation process. So here we are talking about specifically transcription and it related modifications. Okay, first we talked about different splicing mechanism, post transcriptional modification and all those. After that we move towards non coding RNAs. Okay, so first all we all know that there are three RNAs which are very important and present in each and every cell. That is your mRNA, that is messenger RNA, tRNA, transfer RNA, and rRNA, that is ribosomal RNA. Okay, so these are called housekeeping RNAs. Other than that, there are non-coding RNAs like miRNA, microRNA, siRNA, and other RNAs are present, which has very essential role in different kinds of metabolic processes. For example, we talked about doses compensation, right? When two X chromosomes are present in a female body, so that we need to compress that one chromosome, so that the female won't get any double products for the from the X chromosome. Okay, because male has only one X chromosome and one Y chromosome. Okay, other than that, dosal compensation, we talked about imprinting, where non-coding RNAs play a vital role. So this non-coding RNAs specific towards different cell, and they regulate different metabolic processes, which is very important in a lifetime of a particular organism. Okay, so this thing we discussed. Now today, in our today's class, we are going to discuss about telomere and telomerase enzyme. So what is telomere? These are the end points of a particular DNA, which we can added by the telomerase enzyme. Okay, this telomere repeats, and telomerase enzyme helps in resolving the end replication problem. So what is end replication problem, what is telomerase, and how it is related to aging and cancer, and different things we are going to discuss today. Fine, so let's start today's session by this question: what is the sequence specific to telomere repeats? Okay, you can write the answer in the chat box, then we will discuss the uh, answer of this question. Okay, I think that you have already gone through different videos available in your NPTEL site for week 11, so that it is quite easy for you to answer. You can write your answer in the chat box, then we are going to discuss about this question. Yes, if you have any doubt that you cannot answer directly, if you are having confusion between two options, then you can write or guess it, right? No problem. Okay, then let's discuss about it. As you have all said that answer is B. So let's see what is it. Okay, so let's discuss about it. First thing, what are telomeres? Telomeres are basically your repetitive DNA sequences. Okay, at the ends of all human chromosomes and other eukaryotes. Fine. And it was first described in 1975 by Elizabeth Blackburn from Salk Institute of Biological Studies, who recently 2009 earned a Nobel Prize for her discovery. So what they contain? They contain thousands of repeats of 6 nucleotide sequence, that is TTAGGG, 3 guanosine repeats of 11 kb at birth to 4 kb in old age. So why it is written 11 kb at birth to 4 kb in old age? Because you know, every time the cell divides and every time the DNA replicates, the telomere length becomes telomere length becomes shorter and shorter. Okay, because of that, this length of this telomerase repeat, which is 11 kb during the birth of a particular birth of an infant, it becomes 4 kb in the old age. And after that, when the telomere repeats become very short, the cell moves towards senescence or aging, and that cell dies. In that manner, person generally ages. Okay, that's why telomere repeats are connected towards aging. We will be discussing in detail about everything. So this is just a few points that you can remember, that generally it is about 11 to 12 kb, which is a repeat of DNA sequences which is present at the end of all human chromosomes, and it becomes shorter and shorter, so that in old age it reduces its length to 3 to 4 kb. Understood? So in humans there are 46 chromosomes and thus 92 telomeres and each end. And the telomere protect the chromosome from the replication related loss of vital genetic information. So during the replication in the lagging strand, where if there are no telomeres will be present, then some part of the DNA will get cut, and in that manner a vital genetic information will get lost, and we don't want that. So that telomeres are generally present and they cap the DNA chromosome ends to protect the vital genetic information. Fine. Okay, so this is the just we are moving slowly and slowly. So this is the first thing about telomeres. What is telomere? These are generally repetitive DNA sequences, and they are specific sequences, that is TTAGGG. Okay, so here is your answer that you said B. Okay, fine. Let's move toward another step. So what is the function of telomere? Now we just knew about what is telomere, repetitive DNA sequences, and this is the length, this is the specific sequence. So what is the function of it? It protects the chromosome, it separates the chromosomes, it resolves the end replication problem, or all. I think everyone can answer this question if you go through the options carefully. Please try to answer this question, then we are going to discuss about it. Let's discuss. So you all have answered D. That is all. And here is your answer. That telomeres protect the chromosome. They separate the chromosomes in the cell cycle during the metaphase stage after replication, and after this they also resolve the end replication problem. So why do we have them? First thing, why do we have telomeres? Okay, so we need to understand about the replication problem. This is very important. You should listen carefully so that you can understand why we are talking about telomeres and telomerase enzyme. So this is the strand of DNA. As you can see here, this is replication fork. Okay, and from 5 days to 3 days, this is one strand, another strand is 3 days to 5 days direction. So what is your leading strand? Here it is your leading strand, and in this leading strand, this DNA replication occurs in a continuous manner. As beginning of this strand, there must be one primer is there, right? For example, this is the primer. This is the primer with the 3 days end, 3 days OH group. After that, from this 3 days OH group, this adding of nucleotides will start, and this nucleotide addition then further move forward and like this in a continuous manner. Okay, everyone knows that this is about leading strand, and this adding of nucleotides will be done by DNA polymerase enzyme. Got it? Let's talk about this lagging strand. So what about this lagging strand? Here in this lagging strand, we need a primer here. Okay, this is your primer. Okay, this primer is a RNA primer. Okay, this is a RNA basically, and the primase enzyme is responsible for making the primer. So this RNA primer here, as you can see here, this is the 5 days 3 days end, and we need to start from a 5 days end, because only this nucleotides addition will happen to the 3 days end only. It cannot be added to 5 days prime, because 5 days 5 prime region of a nucleotide has phosphate group, PO4 minus group, but 3 days group 3 days end of a nucleotide a chain has this OH group, hydroxyl group. So from the hydroxyl end, you can start just connecting the or making the phosphate groups again and again. Fine. So here, from this onward, after addition of primer, there will be this DNA polymerase will start replication, and after few points it will stop, and there also another primer will come here. Okay, and likewise there are multiple primers is present inside the lagging strand. At the end, the primers will be removed, but there will be a overhang. So let's look at this clearly. See, here is a clear figure. You can observe it here. Just a minute. Fine. See here. So this is your leading strand. Okay, and this is your lagging strand. Lagging strand having Okazaki fragments, and this red part you can see, these are your primers, RNA primers. Fine. So this RNA primers are having this RNA, and it will move towards the replication fork, move outward. Got it? Now you can see removal of primers will happen. So after removing of the primer, you can see there is some overhangs here at this point, 5 days prime, right? This overhang, why there are overhangs are here? Because at the end of this lagging strand, the primer is there, right? But this primer will be removed by the RNA-H enzyme, but the DNA polymerase needs a template to add the nucleotides, and here it cannot add the nucleotides without any help of any primer, because the primer is already removed. That's why there is a overhang. Generally in different cases, what happens here, in different cells of our human body also, in eukaryotes and different organisms, this overhang will be cut down. Okay, this overhang will be removed. Okay, this part will be removed by the enzyme. This part, this is a DNA overhang, so this part will be removed generally in our normal human cells, it is removed, so that this much DNA will be taken care of from the parents to offspring, or for your cell division, different multiple cells like that. Here also, so that in the manner of removing this overhang, we are losing important genetic information. Got it? So think about it. If this cell division is happening a regular manner, for example your blood cells, RBC, the RBC life cycle is only 120 days, right? So it is just a new set of RBC comes after only after just 4 months. So in just a year you are having a 3 set of RBC cycle. So think about your whole lifespan. So in this case, if just this part of a DNA gets removed each time the cell divides, so this DNA must have, for example, it has 50 nucleotides, but at the end of 50 cycles or 30 cycles, it will have only 5 nucleotides, because all these parts will get removed one after another. But we don't want that, okay? We have important information there, and we don't want to lose this information. In that case, here comes our telomerase enzyme. So here you can see there is a overhang here. So telomerase binds to this 3 days overhang, and which is complementary to the telomerase RNA. Let's talk about telomerase enzyme. So what is a telomerase enzyme? It is a ribonucleoprotein complex. So it has some RNA template here, and it is generally a reverse transcriptase enzyme. So what is the function of reverse transcriptase? It can form DNA from a RNA template, right? So this telomerase enzyme has its own RNA sequence or RNA template. So this part of your, some part of your telomerase enzyme, this RNA template will get complementary to the overhang, and it will bind there. Now you can see here it got complementary, that it can act as a primer, and after that this part of your overhang will get replicated. Here you can see, fine, this nucleotides got added, and you can see this is a part of your nucleotide have been this overhang got extended. See, after that this will move forward, and here you can see this is forming another complementary strand, and this overhang further got extended. That means it has, for example, 5 nucleotide overhang, but now it has about 20 nucleotide overhang. Got it? So after this DLA polymerase, now you can see here that this overhang has this much length of DNA. Now this particular, so here what you are having, the overhang, and it is the RNA template. So at the end what will happen, the RNA primer, for example this RNA primer was removed by RNase enzyme, so like that way this telomere will remove, and this template strand got formed, so that the DNA polymerase extends the RNA primer to synthesize a complementary strand. Here you can see it got already a complementary strand this much, and it has some complementary sequence here, so that it will increase its length likewise, okay, in this manner. Now you got your both strands completed, and this overhang will be cut by the RNase enzyme, okay, this will be cut. But in this manner, when you are cutting the overhang, you are not cutting the genetic information, you are cutting the extended part. Understood? So this TAG, this much is your actual DNA. So here you can see this TAG is here, so I can keep that much, and I can cut this much of extended overhang. Understood? Now you are getting all your DNA genetic information back as it is conserved, and this overhang will be removed. So this is the function of telomeres, and they are helping us to protect the DNA or chromosome ends. Understood? Is it clear? Anyone want me to repeat it? Okay then, see. So what do telomeres do? They protect the chromosomes. They separate one chromosome from one another in the DNA sequence, And without the telomeres, the ends of the chromosome would be repaired, leading to chromosome fusion and massive genomic instability. Fine. So how do they got there? This is the telomerase enzyme. You can see here, this is the end part of your chromosome. So this is telomere. Telomere is what? Your repetitive DNA sequence. And you know what is the specific sequence it is. It can be of 10 to 12 kb in case of a baby, but when we get older and older, the sequence gets shorter and shorter. Fine. So this telomerase enzyme has this RNA template, and it acts as a reverse transcriptase so that it can form DNA from RNA. Understood? So if someone asks you what is the telomerase, so telomerase is a ribonucleoprotein complex, because it has both RNA. RNA has template and protein as the function of enzyme. So this is the ribonucleoprotein complex. Understood? Fine. So this is all about your end replication problem. So in the case of lagging strand, we are getting overhangs, and these overhangs we need to comprehend it so that the important genetic information will not get lost. To to manage this end replication problem, we here this telomerase enzyme add the overhangs and just remove the extended overhang at the end. Okay, in this manner we are just managing the replication problem we are having. Fine. Okay, so here is your next question. Which enzyme is called as cellular immortalizing enzyme? DNA polymerase, ligase, telomerase, or P53? Answer this question so that we can discuss it after 2 minutes. Okay, let's discuss it then. I see that you answered telomerase. Yes, it is the correct answer. Fine. Which enzyme is called as cellular immortalizing enzyme? Why it is called immortalizing enzyme? Hmm? So earlier we were discussing about the end replication problem. So when there is a normal cell is there inside your human body, after a replication, the overhangs are there at the DNA end, and this overhang got cut cuts down by the enzyme so that the complete DNA sequence can retracted. Okay, generally. So after that, in this manner this 11 kb of your telomere repeats gets shorter and shorter, and when it becomes so much shorter like 3 kb, 2 kb or 4 kb, so this is the thing where we see the senescence of a cell, that is aging of a cell. Got it? So um, for when in any cell, when there is this bunch of telomerase enzyme is there, and this telomerase enzyme is highly active, that means what will happen, this telomerase, every time the cell replicates, telomerase enzyme will add some telomere repeats so that it will get the whole DNA sequence back. Like that, in different cancer cell, different stem cells, we are getting the whole DNA sequence back using the telomerase enzyme. Okay, that's why it is called cellular immortalizing enzyme. For example, if you want to do an experiment and you take this telomerase enzyme in a particular cell, okay, and you put the telomerase enzyme with a cell, and you see that after a replication cycle of many times of replication, you can see the same DNA sequence you can observe after, even after 20 cycle or 30 cycle. Okay, this has been experimentally proved. So in that case, the cell is never going to die. Okay, this is the key to immortalization, you can say. But not like people can live for 500 years, 1000 years or like that. But yes, it can extend your lifespan from more than 100 years for to add more years. Okay, so this is a immortalizing thing which biology can afford, or which biology is suggesting. Fine. So telomerase is a ribonucleoprotein complex. Why it is ribonucleoprotein? It has this RNA part, RNA template, and a protein which acts as an enzyme. So this can be combined to produce the ribonucleoprotein complex. This is a cellular reverse transcriptase. Okay, that has been referred to as cellular immortalizing enzyme. I think everyone is clear about immortalization, why it is called immortalization. Same thing happens in cancer cells. So in cancer cell, what happen? So everyone knows that cancer cells means they just pass the senescence stage, or they just pass the apoptosis stage. They just keep on dividing, dividing, dividing. So think about it. If there is a short telomerase amount will be less in that particular cancer cell, so the after a number of cell division, the telomere repeats are going to be shorter and the cell is going to die, right? But this cancer cell surpass the death of this cell by increasing, or somehow just increasing the amount of telomerase enzyme. They just activate the telomerase enzyme, they increase the amount, so that even after a longer much amount of division, for a like few cycles, more than few cycles, you are getting the same DNA back. Understood? That's why it is called a immortalizing enzyme. Okay, so it stabilizes the telomere length by adding hexameric repeats, that is TTAGGG, 3 guanosines, onto the telomeric ends of chromosomes. So what is the telomeric end? At the end of chromosome. For example, this is the chromosome, so this will be your telomere end. Okay, so these telomeres are repetitive DNA sequencer, and these are the telomere ends, thus compensating for the erosion of telomeres that occurs in its absence. Understood? So when there is no telomerase, this sequence, this extra sequence is going to reduce its length. So when there is, for example, 11 kb of your telomeres, so this sequence is going to reduce, it will not affect the cell. Next cycle it is going to reduce, it will not affect the cell. But when it affects the your actual DNA, that is actual genetic material, then the cell is going to senescence or aging. Understood? So the telomerase activity is observed in fetal tissue, adult germ cells and tumor cells. It is very important if you just remember this, that fetal tissue, why it is there? Because fetal tissue, here the stem cells are trying to divide and produce different types of organs inside a fetus, right? If we move towards developmental biology, there is different types of morula, gastrula, blastula. These stages are there, where after the epiblast cells or the germ, these cells will move towards the organ formation of a uh, baby. So in this case, this fetus needs to get more blood vessels, more organs, so that it, and what is a stem cell? The stem cell is that cell which can produce different, that is a pleiotropic effect, it can produce multiple types of organs and tissues. So in that case, this telomerase activity is very important. Also the adult germ cells, okay, and your tumor cells. If we don't have telomere in adult germ cells, then when you get a ovum, for example egg cell or sperm cell, in that case the your genetic information will be lost. Okay, some part of your chromosome will be lost. Okay, obviously there is meiosis, crossing over, recombination, everything is there, but still when replication has been happening in the S phase, so some part of the genetic information will get lost, and we don't want that. That's why adult germ cells have the telomerase activity. Understood? And also the tumor cells, we talked about how tumor cells get surpassed this apoptosis and death of their cells by activating the telomerase activity. Okay, and activity is nearly undetectable in somatic cells. Okay, that's why we eukaryotic organisms move towards senescence. We humans age. Why we age? Because of this, because of the somatic cells do not have any telomerase activity. Understood? Okay, so let's see which cell has high telomeric activity: cancer cell, germ cell or stem cell. I think we just discussed about this, so everyone can answer this question. let's discuss it. So, which cell has high telomeric activity? As you have all answered, someone said C. So, all of these cells has high telomeric activity. Okay. So, why all of these cells are high telomeric activity? If you don't get it, so I am just repeating it, that when we are talking about cancer cells, this cancer cells has this specific activity of cell division in a faster mode, so that they can divide in a continuous manner. Okay. And generally, if there is no not much high telomerase activity, the cell will move towards senescence, that the cell will definitely die after a certain amount of cell division, right? But when there is high telomerase activity, so it will just add the telomeric repeats at the end of the chromosome, so that the actual copy or actual genetic information will be conserved, it will not be lost. And in that manner, the genetic information is transformed from one cell to another cell in a conserved manner, so that the cell is fine and it is not going to die. Okay. So that's how the cancer cells surpass the cell death. Next is your germ cell. So these germ cells, adult germ cells, when we are talking about your egg formation or sperm cell formation, meiosis and everything happen. But when there is high telomeric activity is needed, otherwise some important genetic information will be lost, and we don't want to lose any genetic information which will be transferred to the offspring. Okay. Next is your stem cell. Stem cell has a pleiotropic effect. It can produce multiple types of cell or tissues in the fetus. Okay. And this stem cell is also present in your adult stage also, and these particular cells generate different multiple cells. Got it? In that case, there are there needs to be have some high telomeric activity, so the important information will not get lost. Okay. So these all these cells have high telomeric activity, just remember this. Or you can, I am just trying to let you understand the whole thing, so that it will be easy for you to get the point. So how does this telomerase work? Research also shows that the counter that controls the wasting away of telomere can be turned on or turned off. Okay. So this wasting away of telomere, that means, for example, this is your actual copy of DNA. After replication, this will be this much. Because of the overhang, some part of the DNA has been lost. So this wasting away can be turned on or turned off. The control button appears to be on telomerase, which can rejuvenate the telomere and allow the cell to divide endlessly, which happens inside your cancer cells. Most cells of the body contain telomerase, but it is in off position, right? So inside your somatic cells, you have telomerase, but it is in off position. Telomerase is not active. But when you get some part of your body has some cancer, even if it is a somatic cell, it will have the telomerase activity. Okay. So that the cell is mortal and eventually dies. Understood? Because of that, the cell eventually dies and people move towards senescence, aging. So telomerase works by sorry adding back to telomeric DNA to the ends of chromosomes. So what is telomeric DNA? They are the repetitive sequences, thus compensating for the loss of telomeres that normally occurs as cell divide. Okay, let me explain it for you if you have any confusion. For example, this is your chromosome. Okay, and here you are having this telomeric repeats. This is your telomeric repeat. Okay. So this is your chromosome. For example, this is a chromosome 1 or chromosome 2, whatever it is, you can just consider it. And here you are having, this is your DNA copy, okay. This is in 5 days to 3 days end, and this is in 3 days to 5 days end. And these are your telomeric repeats. At the end of each and every chromosome, you have telomeric repeats. So don't need to confuse about that. So when the cell divides and it moves towards the end of replication, some part of your telomere will be cut. So now it will be like, this is your chromosome, okay, and you will have telomeric repeats like this. Now you can see that the length of the telomeric repeats have been shortened, okay. For example, this is of 10 kb, but now it is of 9 kb. That means 1 kb has been lost because of the end replication problem, that is the overhang. Because of the overhang, we need to cut the overhang so that you can get the whole copy of DNA. In that manner, you are getting a lesser copy of telomere repeats. This is 10 kb of telomere repeats, now you are getting 9 kb. In that manner, after a longer cell division, it will be reducing. So when there is telomerase activity, this is the enzyme telomerase, okay. This is the enzyme telomerase. When it is active, so what will it do? It will just add the telomeres back, okay. Here your telomerase is adding your telomeric repeats. Now you have lost 1 kb of sequence from the cell division, now you got it back. So in that manner, after the cell division, you are having 9 kb, then also the telomerase is adding its telomeric repeats. So in that manner, you are having your 10 kb of your telomeric repeats, it been just same after a number of cell division, okay. That's why the loss of telomeres normally occurs when cells of divide, and in that case, when there is telomerase active, it just add the lost telomeres. Understood? Is it clear? And that's why in cancer cell and germ cells, the genetic information is same, and the telomere repeats is not going to sort, so that the cells can divide endlessly, continuous manner. But normal cells like our body, somatic cells, okay, they do not have this telomerase enzyme, okay. Like our skin cells, like your muscle cells, skin cells, they do not have this telomerase activity. So when I am at the stage of age 20 or 25, but when we move towards age of 50 or 60, then my skin cells were going to age, okay, the wrinkle formation will happen. Why there is wrinkle formation? Because the like some loss of DNA happens, and in that case, this lost cannot be repaired further, okay. That's why we are facing aging and senescence. We are talking about somatic cells, regular somatic cells. Understood? Is it clear to everyone that how telomerase is working in case of somatic cell, and in case of germ cell, and in case of cancer cell? So in humans, telomerase is active in germ cell, in vitro immortalized cells, the vast majority of cancer cell, and possibly in some stem cell, okay. High telomerase activity exists in germ cells, stem cells, epidermal skin cells, follicular cells, and cancer cells, okay. So you can say that telomerase activation or telomerase, presence of telomerase is the hallmark of cancer, okay. For example, you are getting a tumor tissue tested, that you are just extracting a tumor from a mouse model, and you are observing and just testing it, experimenting on it, and you found there is some telomerase activities there, presence of telomerase enzyme you found. That means it is a hallmark of cancer. That means there is some cancerous cells are present in this particular tumorous environment. Understood? Next is, some cells are immortal because their telomerase is switched on. I think it is clear to everyone. If you have doubt regarding this, you can ask me. Okay. So the cells become immortal when this telomerase is switched on. An example of these immortal cells are blood cells and cancer cells. Why blood cells? Because these blood cells are formed from the bone marrow cells of your body, and these bone marrow cells, uh, they need to be have some telomerase activity so that important information cannot be lost. And the as I told just a few minutes before, that the RBC, the lifespan of RBC is only 120 days. So even after just 4 months you are going to get rid of those old RBCs and you are getting new RBC. So in that manner a lot of cell division going to happen. So think about it, if you just in a year you are getting 3 sets of RBC, so in your whole lifespan you are getting how much sets of RBC? Okay. So in that case, if you don't have telomerase activity in case of blood cells, that means that your genetic information, DNA information is going to lost. And if your DNA is lost, that means you are getting malfunctioned RBC, which will lead to different types of cancer and malfunctioning inside the body. Understood? Cancer cell do not age because they produce telomerase, which keeps the telomere intact. As I already told you, that cancer cells do not age, they can keep dividing endlessly because of this telomerase enzyme activation. Got it? Fine. Let's move to next question. So there is a term called Hefflick number. So what is it refers to? Age of a cell, number of times a cell can divide, stage of DNA replication, cellular senescence. What is it? Okay, let's see. So what is it? Heplik number. This is basically a number of times a cell can divide. So it was given by Leonard Hefflick. Cell division is a finite biological function and inexorably tied to be aging process. So this is very important. If you can understand it or if you can remember it, try to understand the thing so that it will be easier for you. So cell division is a finite biological function, it is not infinite. Okay, that every cell of your body has some finite number of times it can divide before moving towards senescence or aging. Okay, except what is the exception? Tumor cell, cancer cell, germ cell, they are the exception. So this Hayflick number describes the number of times a cell can divide. Hayflick was the first to suggest the relationship between cell division and mortality. Mortality means aging or senescence of a particular cell. But the mechanism was not initially understood, it was understood later. So after his name, Leonard Hayflick, this is called Hayflick number. That means a cell, this is the number of times a cell can divide. Got it? Okay. Let's move to next question. Telomere length serves as intrinsic biological clock at regulating lifespan of a cell. Is it true or false? Yes, you can answer it if it is true or false. I think it is quite clear. Okay, as you said it is true, then I think that you understood the concept clearly. It is very good that it is a intrinsic biological clock at regulating lifespan of a cell. How is it? See, first let's just rethink or let's discuss about it again, that what is the end replication problem. So DNA polymerase can only synthesize DNA in the 5 days to 3 days manner, and resulting in a discontinuous replication in the lagging strand with Okazaki fragments. Okay, this is the whole okay, just a minute. Okay, I got it wrong. Fine. So this is your whole strand of DNA. Okay, for example, this is, so in this case, once when you have this 3 days prime end, so here your replication will move like this is the leading strand. But when you have 5 days prime, this replication will move in a discontinuous manner. Why there is discontinuous manner? Because you have small RNA primers at the beginning of this every DNA fragment. These are different small RNA primers. So what are these RNA primers? They are essential for the DNA polymerase to add the nucleotides. Got it? So but at the end these RNA primers will be removed. Okay, why they will be removed? Because it is a DNA replication and we don't want RNA primer here. So this RNA primer will be removed by your enzyme called RNase. This is your RNase enzyme which remove this RNA primers. So at the end of this you are getting your DNA like this. Okay, this is your leading strand, Now you see your lagging strand. How is it looking? Like this. Because your RNA primers have been removed. Now you can see at the 5 prime end, sorry, at the 5 prime end, where there was RNA primers are already there, this green color part, this part is empty. Okay, this part is empty. So this is called a overhang. Now this overhang is the problem. This is called end replication problem. How generally in case of our somatic cells, this overhang is removed, but in case of our other cells like germ cell, cancer cells, we cannot remove this overhang. So consequently DNA synthesis does not extend to the very end of the lagging strand, right? In the absence of a protective mechanism, the end replication problem means vital genetic material from the end of lagging strand would be lost during each cell division. That means this part is lost, and telomeres solve the end replication problem by protecting the cell from loss of its critical coding base pairs. So now if you have this telomeres here, this part will be cut by other enzyme so that only the telomeres have lost, not the genetic information. Okay. So limited capacity of cell to replicate. The cells have limited capacity to replicate. They cannot replicate for a like endlessly manner, because it has only a finite number to replicate. And the telomere length serves as a biological clock. In the beginning of this session, we talked about that 11 kb is the whole length of a telomere repeat in case of an infant or in case of a baby. But when we get older and older, the length of telomeres uh, decreases, so that if we get a cell or bunch of cells where we get the length of telomeres, so how much it is shorter or longer, depending upon that we can predict the person's age. Fine. And Haplik limit is the maximal number, not a minimal, for cell division that can achieve in vitro. And when cells reach this limit, they undergo morphological and biochemical changes that eventually lead to arrest of cell cycle and process called cell senescence. Understood? This is called cell senescence. Fine. Let's move to next question. How telomeres affect the quaternary structure of DNA? Adding a base pair, epigenetic modification, steric or electronic effect, promote recombination. What is it? telomeres may affect all the four structural levels of a DNA, that is primary and secondary structure. They protect the integrity of base pairs and preserve basic genetic information, right? We talked about it. Tertiary and quaternary structure, as telomeres shorten, they may exert different steric and electronic effects, okay, resulting in changes in the shape of DNA. Fine. So in case of tertiary quaternary structure, what is it? It is when the DNA bind to its histone molecules in the chromatin or compacted from in case of chromosome. In this case of compaction is there. So when that is called your tertiary or quaternary structure. So in that case, when the telomeric length shortens, it may affect the shape of DNA by causing different steric or electronic effects. These changes may in turn affect the genes exposed and available for transcription. For example, if your length of your DNA is 10 kb, okay, this is your 10 kb length of DNA. When you add the histone molecules here, this is your histone molecule 1, histone molecule 2, so it will be wrapped around it, right? It will be wrapped around it like this. So this part of your DNA and this part of your DNA is present for transcription, and this part of DNA which is inside of this histone molecules, it is not available for transcription. Got it? Now when you just cut some part of these telomeres have been sorted, that means this modification will change. Okay, how it will change? Like this is a histone molecule here and this is another histone molecule here. So here your DNA replication, DNA will move like this. Okay, for example, this part of your telomeres have been sorted out. That means it will affect the genes which is exposed for transcription. Understood? Here it is the length, that means this part is cut, this much part is cut because of your cell division. That means this particular length of DNA is need to rearrange as comparison to before histone molecules. So what do? They generate more than a genetic clock. It will help you find out the age of a particular cell. That's why it is called genetic clock or biological clock. Protective cap, it produces a protective cap at the end of your chromosome and protect chromosome from recombination, exonuclease degradation and end-to-end fusion. Okay, end-to-end fusion, you understand that one end of a chromosome is interacting with other end of a chromosome because there are only nucleotides and nucleotides can form complementary base pair, that's why. And also protect chromosome from recombination, exonuclease degradation. What is the meaning of exonuclease degradation? So what are endonucleases? Endonucleases are the enzymes which cut a DNA at a specific sequence inside the DNA length. Exonucleases are the nucleases which cut the DNA from 5 days end or 3 days end. They don't cut at the middle of it, so that when there is a cap, so that it can protect it from the exonuclease enzyme. Distinguish telomeres from DNA double strand breaks. Okay, that would hinder progression into the G2 phase and inappropriate recombination events. So it can distinguish the telomeres from the DNA double strand break. When there is a DNA double strand break, so that it will distinguish it. If the like for example, if your a cell is moving towards cell division, and after a completion of cell division, the length of telomere will shorten down in case of a somatic cell. But when there is a double strand break, so it will not complete its own replication. Okay, because after double strand break there needs to be non-homologous end joining and all like that, different types of DNA repair mechanism are there. So it will just distinguish it and it will help the distinguish the DNA so that it will not move towards the G2 phase. Okay, and prevent oncogenesis, as we have talked about it. If telomerase is not active, cell will move towards senescence and it will not just repeat its division endlessly, so that it can prevent oncogenesis. Got it? So this is about the function of telomerase enzyme and telomere repeats. Okay. So here is another question about chromosome compaction, as we have just discussing about this DNA, histone and quaternary structure and all this. Which of the following cause chromosome compaction? PRC2, snRNA or shRNA or HAT? I think you can understand its full forms, because we have already discussed about this uh, RNA, different types of RNA. Which one is a non-coding RNA, which one is a matlab regular RNA. We have discussed about it. Okay, so you can answer this. Yeah, I'll tell you. This is steric and electronic effect. Okay, this is steric and electronic effect which affect the telomeres, uh, which caused by the telomeres, which affect the quaternary structure of DNA. Fine, then let's discuss about this question, which is you already have answered, that is PRC2. So what is PRC2? Let's see. So this is called this is your DNA. You can see it is in black color, and this is your histone molecule, which is in sky blue or cyan color. These are kind of disc like structures are here. So this DNA length has been wrapped around these histone molecules to form a beaded structure. Okay, and for what helps in this process, this PRC2 and PRC1 complex, there are different types of molecular complexes are here which helps in this wrapping process. Now you can see this is your length of DNA which is exposed, but after this PRC1 and PRC2 involvement, the length has been clearly been compaction been happened. Okay, so this compaction is achieved through the wrapping of DNA, around certain proteins, the histones forming the building blocks of the chromatin fiber, the nucleosomes. Got it? So here histones are forming the building block around which the DNA wraps to produce the nucleosomes. And this PRC2, which is a polycomb repressive complex, is a conserved chromatin regulator that is responsible for methylation of histone H3 lysine 27, that is H3K27. If you remember it correctly, so we have already discussed about the chromosome compaction while we were discussing about doses compensation. So in case of doses compensation, what happens? This X chromosome length decreases and it becomes shorter and shorter like a small fragment. So because how this higher compaction happens? Because of this methylation process and acetylation process. This methylation acetylation happens in the histone, and these are called epigenetic modification. So this H3K27, if you remember, is highly responsible for chromosome compaction. Okay, so this methylation and deacetylation process responsible for chromosome compaction along with PRC2. It is essential for normal development, and its loss of function thus results in a range of developmental phenotypes. Okay, so this developmental phenotypes can be achieved through this process. Got it? So let's move to next question. Telomeres can be visualized using fluorescence microscopy, real-time PCR, northern blotting or western blotting. Okay, so here is the answer as you have already answered. Very good. So you can visualize it using fluorescence microscopy. Okay, fine. So see, we have already discussed about telomerase enzyme, telomere repeats, aging, senescence, cancer cell, everything. So we are going to see the questions. If you have any doubt regarding this, you can ask me. Okay, so try to answer this. DNA end replication problem is elevated by RNA polymerase, reverse transcriptase, telomeres and telomerase. Which one is it? Yes, answer this question. have already discussed it so many times. So DNA end replication problem is elevated by telomerase enzyme. Okay, there are telomeres. They help in just elevating the replication problem, but for a certain amount of time. But telomerase, if there is telomerase activity is there, it will help you to elevate the problem in case of your germ cells, stem cell, and epidermal cells, follicular cells. They are essential where there is telomerase activity is on, and it elevate the replication problem. And some of you have said reverse transcriptase. See, reverse transcriptase is the enzyme. Telomerase enzyme has activity like reverse transcriptase, but you cannot say reverse transcriptase. For example, if I just put a HIV reverse transcriptase, so that it will help the elevate the DNA replication problem, is it? Always try to understand that telomerase enzyme, it is a ribonucleoprotein complex. It has some RNA template already present. It has some RNA template of its own. Okay, and other than that, it has this protein which has the enzymatic function like reverse transcriptase. Got it? So that using that RNA template which is already present inside the telomerase enzyme, the end replication problem can be resolved. Understood? Fine. So here now you can answer this. Telomerase come under the enzyme category. Nuclease, reverse transcriptase are DNA polymerase and DNA ligase. Okay, let's see. So as you have already answered reverse transcriptase, so this is the answer. Okay, so let's little bit discuss about these enzyme categories. What is nuclease? Nuclease is the enzyme which can cut the phosphodiester bond between two nucleotides, and it can be of endonuclease and exonuclease. Endonuclease cuts the DNA from inside with a specific sequence, uh, where it has some specific sequence. It can cut there only. Okay, so and exonuclease, this as the name suggests, it can start cutting from a one end like 5 days end or 3 days end, whatever it is. Fine. Next is your reverse transcriptase, that is can form DNA from RNA. Third one is your DNA polymerase, which is essential for DNA replication. DNA ligase is can join two strands of DNA, so that it can form the phosphodiester bond. Okay, So cancer risk is high when telomerase is inactive, telomerase is short, telomerase is active, or P53 is active. Which one is it? We have really discussed a lot about cancer cells, so I think you can answer this question. Read the question again and answer it. Okay, so I think you answered it C. Great, so you have answered it correctly. So when telomerase is active, it just keep on adding the telomere repeats at the end of your chromosome, so that the telomere length will not get shorten and shorten after each cell division, and it will help to retracting the length of the DNA after every cell division, which will increase the lifespan of a particular cell, and it will not going to die or moving towards senescence. That's why the cancer risk is very high when there is a active form of telomerase present. You can call it as a regulator of tumor cell. Next is telomerase is a repetitive sequence present anywhere on DNA. Is it true or false? Just these are some basic questions, conceptual. Okay, let's see that you have answered false. So it is correct. Okay, because telomere is a repetitive sequence which is present at the end of DNA at the as a cap. Okay, so it generally helps in capping of the chromosome. so that the ends will not interact with each other, and it will help in the DNA replication problem. Got it? So telomere length is maintained constant in all chromosomes. Is it true or false? Fine, very good that you have answered correctly. It is also false because it is not maintained constant in all chromosomes. In case of your germ cells and stem cell and other cells, it just the chromosome length is maintained, but in your somatic cells, the length of telomere decreases as you old. Okay, as the person old, so that age increases, so the length of telomere decreases, and that is the mark for your genetic clock or biological clock. Or you can say that if it, the what is the age of that particular cell, if it is moving towards senescence or not. DNA end replication problem is never seen in stem cell. Is it true or false? It is saying that end replication problem is not there in stem cell. Yes, answer this question. Great. So this end replication problem is there in the next stem cell, but because of the activation of your telomerase enzyme, it is resolved. Okay, it will not a problem for stem cell because the telomerase enzyme just keeps on adding the telomere repeats at the end of chromosome, so that it will not shorten for a time being. Okay, so this is the last question of today's class. So cellular senescence is not seen in all cell types. Is it true or false? So it is true because it is not seen in all cell types. It is only seen in your somatic cells. Fine. Once the telomere shrinks down to a certain level, the cells can no longer divide and its metabolism slows down, it ages and eventually dies. Healthy human cells are motile because they can divide only a finite number. You can call it as a Hayflick number. Okay, growing older each time they divide. The cells in an elderly person are much older than the cells in an infant, and you can find out it what is the age of that particular cell using your or you can see the telomere repeat, what is the length of this telomere repeat is present inside a particular cell, so that you can find out its age. Got it? So this is how telomerase enzyme helps in your end replication problem in and also it is connected towards aging. So aging, senescence and cancer cell, how they just keep on passing towards the they just keep on passing the death or apoptosis of or cell death, right? So how this happens, we already discussed about everything linked towards telomerase and telomeric repeats. So thank you all. If you have any doubt regarding this session, then you can ask me. Hayflick number refers to the number of times a cell can divide. Okay, every cell has a finite number of times it can divide. Okay, just a minute, I have a slide for this. This is called Hayflick number and it is given by Leonard Hayflick. It describes the number of times a cell can divide. So if you see, for example, this is your chromosome, okay, and this is your telomere repeats. This is your 10 kb of telomeric repeats. So after your, I am talking about a somatic cell. After a certain number of cell division, this telomere repeats will be decreasing its length. Okay. When it moving towards 10 kb to sort of like 1 kb or 2 kb, that means the length of the telomeres are decreasing, so that and eventually it will affect the DNA also. So when it affects the DNA, that means your cell is going to die. When there is a DNA malfunction, that will lead to production of malfunction proteins and eventually the cell will die. Okay, so that's why every cell of your body, that is your somatic cells, is motile, that is it is going to die one day. And that how many number of times it is going to divide, it is given by this Hayflick number. This is called maximum number of times a cell can divide. Got it? Is it clear?
Yes, ma'am.
Okay, great. Anything else? Okay, fine. Thank you everyone for joining. We'll see you on next class on next Monday.